Hi all. Welcome to people online as well. Hopefully you can hear me. Today we have a special guest, Dr. Viviana Gonzalez Astudillo. And I think actually this is particularly special because you sought us out. A lot of times we're chatting with friends or people that we know, people we've been working with. But Dr. Gonzalez Astudillo came to us and said, I'm just back here. I would like to be involved. I would like to know what people are doing in this area. So thank you very much for your seeking us out. That's great. Her talk today is from Microscope to Megafauna, a veterinary pathology's role in wildlife conservation. And I'm gonna give you a quick bio. So about Viviana. Viviana is a board certified specialist in anatomic pathology, which just sounds scary to me. I don't know about anybody else, so you might actually have to describe what that is, so people don't move further away in the room here. Working as a lecturer and a clinical academic at the University of Queensland in the School of Veterinary Science. She was born in Colombia, one of the mega biodiverse countries in the world, a reality that has shaped both her professional and personal life. Something we have in common is that we both spent time in higher education in Georgia, which makes us like a select very few people in the world. We've chosen that wonderful state for such things, but actually the University of Georgia is an excellent institution in terms of conservation and has a lot of history in conservation and wildlife management. And then did a postdoc back here looking at pathology of koala diseases. And upon finalization of her doctoral work, she completed her anatomic pathology residency at UC Davis, which prepared her to attain her board certification as a veterinary specialist. Given my troubles with ethics permits with animals, I think you may be my new best friend. We'll be talking in depth about how to get through ethics permits, animal ethics permits, that is. Okay, sorry, without any further ado, Gonzalez Astudillo.
Please, Viviana. Please call me Viviana. I feel that's a very long surname. So, anyway, I don't have to really reintroduce myself, but I did want to say thank you so much to Daniel for hosting the morning team, for inviting me here, and also to Kate for organizing everything. I've received all the very specific details. This was preparation for this. We just went very smoothly, so I appreciate that. Oh, sorry, this is off. So I did create this little Padlet exercise. So if you can please pull out your phones, those of you, this will be a completely anonymous survey. The goal is that as soon as you use the QR code, it's going to take you to a Padlet page, and it'll ask you a simple question, which is either, can you please share a cool fact about veterinary pathology, or what do you know about veterinary pathology? So I just want to know, get a little gauge of how people are in the room about my career. So this will also be at the end in case some of you just don't get a chance of scanning it right now. Oh, good. So I was going to say all this, but I probably can skip it now since Daniel has covered it. But I was born and raised in Colombia. So Colombia is just in this country up here in the northwestern country of South America. And we are really blessed with this particular location because we have a lot of biodiversity. There's a really incredible gradient in landscapes in the country. We have the Amazon here in the south. We have eastern plains with lots of grasslands in the east. We have the Choco biogeographic region. Some of you may have heard that term before, but it's one of the highest levels of biodiversity per square mile in the world. We also have the Pacific Ocean, we're bordering on that side, and the Caribbean Sea in the north. We also have deserts in the country. So the fact that we are one of the 7 mega diverse countries is quite remarkable considering the size of Colombia, compared to a country like Brazil, which is pretty significant. Our national flower is the orchid. The wax palm tree is our national tree. The condor, the Andean condor, is the national bird. And I've been really focused about, since I was a little girl, I was very passionate about animals. My dad used to just, give me that input of, if you see a little bird, let's try to take care of it. So for me, it was very clear that I wanted to become a veterinarian since I was, a small child. And after that, Colombia is the #1 country in the world for, avian diversity. So we have the most number of bird species in the world. So it's, that obviously shaped not only who I am right now, but also my career and where I really care about. So now that I'm working in the vet school as a specialist, this has opened an incredible amount of opportunities for me to, talk and basically make my dreams since childhood come true. So I don't know how many of you actually know pathology, but the actual word pathology comes from 2 separate words from Greek. Pathos means suffering, and logia means the study. So it's literally the study of diseases and what causes those diseases, pathogenesis, so all the way from a cell getting injured by any particular agent, infectious and non-infectious, all the way into that actually creating disease, and the resulting structural and functional changes that come from the generation of disease. So I am an anatomic pathologist, and as Daniel said, I will get into this explanation. So veterinary pathology is split into two worlds. We have anatomic pathologists and we have clinical pathologists. Anatomic pathologists, which is what I do, are the ones that do cadaver dissections, and we look at the microscope, for those tissue examinations once we collect them from a cadaver. We also work with living animals, so we also look at surgical biopsies. And in the human world, it's the same thing. Clinical pathologists are the ones that work with bodily fluids, so they receive samples from, let's say, blood serum, blood plasma, urine, saliva, etc. And so I will just be talking about anatomic pathology today. You may have heard the terms in general postmortem examination, so that's what I do in my career and when I teach the students in the vet school. And some of you may have actually heard the terms necropsy and autopsy used in a very different manner. So what I was taught, which is not correct anymore, is that necropsy is for animals and autopsy is for humans. If you just please remember one thing from my talk today, just scratch that. You can safely use the word autopsy for any single species in which you do a postmortem examination on. And that is because autopsy comes from the word autopsia in Greek again, which means the act of seeing with one's own eyes. It has nothing to do with the species that you perform an examination on. So, also carrying out autopsies takes a lot of technical details. There's, it is from the containers that you're going to put your samples on. What's your dissection approach that you're going to do in a particular animal according to its anatomy, documentation, photographic records, tools, all of those aspects I'm going to leave out of the talk, obviously. I just don't have enough time, and that's just a whole world on its own. But I'm also going to leave here, and this will be recorded, so it will be available for everyone. There's heaps of resources online, and also, by probably contacting institutions, you can even probably get a printed copy or buy a printed copy. But there's heaps of resources online about handbooks, dissection approaches, especially from the Association of Zoos and Aquariums, the American, the World, and the European one. We also have the IUCN, so the International Union for Conservation of Nature, also providing some of those resources. And even going a step further, if anyone is interested in doing dissections on actual autopsies and histopathology in invertebrates, there's also now resources for that, which was quite remarkable for me. I found this out during my residency, and I was pleasantly shocked, because you can do dissections. It's very oriented in invertebrates that are production animals, such as honeybees or silkworms, but the resources there. So. Despite the fact that I really love doing wildlife autopsies, it comes with challenges, and all of you work with wildlife, so obviously you're very well acquainted with this reality, and that also affects what we do in my daily job. But the exercise is just so rewarding. You never know what you're going to find. So to me, every single case becomes like this little fascinating investigation that I really want to unravel. So one of the first challenges that I think it's a really important one is the fact that functionality issues are not going to be the same as morphological changes. So in other words, if I have an animal that is severely affected in front of me, let's say like a neurological bird or neurological flying fox, I may be, very encouraged to think, oh, I'm sure if I just, get that animal once it dies and I do an autopsy, it's obvious that I'm going to find what's killing the animal, what is causing that severe disease. And it's a reality that you just open those animals and there's nothing. And then you say, well, maybe the microscope is going to be something, and there's still nothing. So that is a reality with domestic species, even with humans, but obviously it happens with wildlife. So we just have to deal with that challenge. And that actually became quite clear to me when I started working very recently with these two paralysis syndromes that are affecting our Queensland wildlife. And we're about to enter, or actually we've already entered because it's already quite warm and humid, the lorikeet paralysis syndrome. So that is affecting thousands and thousands of numbers of rainbow lorikeets. These animals come to you, they can be completely unable to fly. They have these really horrible voice changes. These animals if they're just left out there in the wild, they're going to die. So if you take them to a veterinarian, they're severely affected. If you do autopsies, as it's been done already, there's nothing in their tissues. There's nothing in the neurological, in the central nervous system, nothing in the spinal cord, nothing in peripheral nerves. And if you do find something, it tends to be like a very incidental finding, maybe like a foreign body penetration, or maybe some crusting or some trauma, external trauma, just from trying to drag themselves, to go to safety. So this led us to be contacted by Wildlife Health Australia, and they are the ones now that sort of like directing or like helping us direct a lot of those efforts. So now we work as a, as a team, not just as individual researchers trying to find causes. So Wildlife Health Australia has sort of like encouraged us to create two separate teams. So now there's a diagnostic investigations team for the paralysis syndromes, and I'm a co-lead in that team. And there's also the emergency response team. So we have epidemiologists, wildlife veterinarians, biologists, wildlife experts, wildlife people that work in wildlife rehabilitation. So all of that team is hopefully, put together as earlier this year to maybe accelerate, us finding these, or what causes, or the multiple causes if there are multifactorial conditions for these two syndromes. Another one is that unfortunately, when you work with wildlife, preservation artifacts are really common, and they come in separately, like in two big sort of groups of preservation issues. When and you guys go out in the field, you do your your samples for your particular project, but you may actually incidentally find, let's say, like 5 or 10 dead echidnas. It will be very nice. That's sort of like working the first line of defense. You will want to, if you have the space and you have the ability to capture those echidnas and then submit them for a postmortem exam, that will be fantastic. So. But you don't know for how long they've been dead. They might look okay on the outside, but these animals are very covered. They have like these really big thorns. So it's really becomes difficult to know for how long they've been dead. And the other thing is, even if an animal dies in front of you or is euthanized and is fresh, if it's going to take a week or 2 weeks to get to the actual place where we'll do our postmortem exam, it might be better for you to freeze it. But then again, that comes with challenges. So freezing is a common thing that we use in pathology. We also use it a lot, with the small animals that get submitted for a postmortem exam. Sometimes it can be simple things like the owner isn't sure that they want to submit an animal for a postmortem exam, and then once they make the decision, once they're finally, their mind has come clear and they say yes, they do want to know what happened, they get to us, and it's maybe 2 or 3 weeks after the time of death. So obviously, always with pathology, the pressure is going to be better, type of quality that we want, and this is. I didn't have an actual picture, a good picture of a wild animal. This is a German Shepherd dog that got to us this year for postmortem examination. So it had a splenic tumor. You can see this really big mass that is completely disrupting the architecture of the spleen. You can't even recognize that's a spleen anymore. And this animal was frozen and thawed. And what we do in Gatton to try to preserve the tissue quality as much as we can is that we do a slow thaw. So we place the animal that has been frozen at -20 Celsius into a big walk-in fridge that is on Fort C. And so we wait about maybe a week and to do that slow thawing process too. And it tends to help us with the tissue quality. So when you do that, when you are freezing animals, you create a lot of these ice crystals, and they're going to rupture a lot, the vast majority of cells, especially red blood cells, leukocytes, epithelial cells. So you get that very, kind of like a very eosinophilic, or maybe like that's an eosinophilic pink or purple soup in the microscope, which you have to learn to see beyond. So when you freeze and thaw, there's going to be a lot of fluid coming out of the animal, and that fluid tends to fill the cavities, and we call that purge fluid or freeze thaw fluid, and that is inevitable. In this case, you can see there's a little bit of fluid down here. Obviously, this is a bleeding mass, so it doesn't really help. But it's very common to see this very watery fluid. It's not really blood. It's not really forming clots. It's just that massive amount of fluid present in the cavity. Another thing that happens is that when you rupture red blood cells, they carry hemoglobin. That's an endogenous pigment that we have in our bodies, and that just stains everything painful red. So this is exactly what you're seeing here. There's a lot of pink discoloration, or like dark, even like maybe moderately purple discoloration of the intestinal loops. And this animal did not have an enteritis. So this is just that preservation artifact. So with experience, and when I was doing my residency, it was basically just discussing it with my supervisor. Is this a real change or not? That just takes years, and you getting acquainted, with the different tissue qualities. Other things that we have with wildlife, inevitably we have too many wildlife species, and the bulk of knowledge in veterinary pathology just goes to our animal species, so companion and production animals. So inevitably we just have to go to that literature sometimes and use that information to help kind of like understand what's happening to anything that we're seeing in the microscope or during our postmortem exam. I've even checked the human literature, because I have found diseases. It happened once in a koala that I found a disease that, or lesions that were really compatible with a very weird syndrome in humans. So it helps. And we have to do this obviously carefully, because extrapolation is not, a direct It's not appropriate to just assume that everything is because we're mammals, we're all the same. Doesn't apply. The second important challenge here is primary versus secondary lesions. You may be driving around and you may be seeing, like, unfortunately, I travel around the Borrego Highway. I do see macropods being killed by cars and they're just sitting in the side of the road. Did that animal just happen to have gone into misadventure because it was dark at night or like at dawn and then it just didn't see the car and got killed? Yes, probably. And a lot of wildlife unfortunately get killed in Australia because they just happen to be close to roads at the wrong time. But that doesn't mean that their animal was fine. So unless we do a postmortem exam, we're not going to find out if that animal maybe had an underlying condition that was making that animal or rendering that animal vulnerable for, to just basically be involved in a vehicle accident. So that's always important to try to understand. And all of you will know very well the last point here. So field sampling, collection, and storage can also be a problem. When you're preparing to go in the field to address an outbreak, you need to have the right type of containers. You need to take some containers that are sterile. You have to have the right media to keep those samples alive if they're involved in pathogens. And obviously, if you need to ensure a cold chain, you also need to have dry ice, ice packs and things like that. So all of those are different challenges. To basically one single project that I could give as an example to all of these challenges is in a research project that I had with the University of Southern Queensland. Incidentally, a PhD student there that already graduated, he was driving around the Lockyer Valley, which is that area around Gatton, and he started seeing dead raptors. So he thought, obviously this could just happen incidentally, but he started seeing them being more normal, higher numbers than normal. So he got concerned and he contacted us in the vet school and said, is there anything that we can do with these bodies? I could collect them. So I said, yeah, let's put in a project and try to get some funding to at least try to do some toxicological analysis in these raptors. So because some of these raptor deaths coincided with the mouse plagues that happened around the Lockyer Valley, we were aware that farmers were deploying anticoagulant rodenticides, and there's first and second generation groups that they can use. And so we started collecting the samples. We ended up collecting 24 raptors in total. Actually, 23 raptors and one nightjar, so one carnivorous bird, but none raptorial. And one of those, this is just to give you, probably this is the best picture that I have. This was a bulbul owl that he collected. And we can see here, this is exactly what makes us concerned about anticoagulant rodenticide, secondary anticoagulant rodenticide poisoning in raptors, is the fact that this animal has bled now into its coelom, which is the cavity in birds that holds its organs, and there's no real clot formation. So when you see that in an animal, that becomes a concern, especially if it's a wild animal. And then if you see the liver, I don't know how many of you have seen livers, but they should be a lot darker red than this, or brown, because the liver is a very vascular organ. So this animal has exsanguinated now into its coelomic cavity. So all you have behind is basically the stroma, the epithelial cells that form the bulk of the organ. And then you have these little things over here, which are petechiae, so very tiny deep dot hemorrhages. So with this case, we were like, oh, we're definitely going to find, these lethal exposures in these guys. We collected livers, sent them for LCMS, so liquid chromatography mass spectrometry analysis at CSIRO, and all of the results came back as, yes, they had been exposed, but likely it was just sublethal. And I say sublethal with a little, parenthesis here, because we don't have the information for what is lethal in our raptorial species. What we have in the literature is information on what is the lethal dose 50 of whales, mallards, and species like that. So when you're trying to establish what level of toxic or a chemical is going to kill animals, you have to do a control experiment. And once you do that, you give them, this particular substance. And then when 50% of them die, that's what you call the LD50. So we don't have information for LD50 for our native raptors in Australia, for the ones in the Lockyer Valley. So using extrapolated information from at least other bird species, we were able to come back and be like, yeah, it's likely it's a sublethal exposure, but it's still important to keep monitoring these animals and that we don't end up with lots of poisoning in our raptor species, because they're so important for the ecosystem. Another one is that especially as wildlife diseases become more, more like common or very obvious in front of our eyes, we need to establish a case definition before we actually go there and try to investigate causes. So, and case definitions are just a way of you to put together a mechanism to call an animal, is it disease or not, for the disease that I'm particularly interested in. So a perfect example of this situation was the flying fox paralysis syndrome and the lorikeet paralysis syndrome, because when animals start becoming neurologic, there's many reasons why animals can be neurologic, but in order for the researchers at the time to start calling these cases lorikeet paralysis syndrome or flying fox paralysis syndrome, you had to get together a multidisciplinary team. So you get together epidemiologists, wildlife veterinarians, ecologists, biologists, and then you come up with this set of criteria that is like, am I going to be able, with this animal that I have in front of me, and I'm going to be able to call it a confirmed case for my disease, probable or possible, or is it just going to be a negative? And that is just the first stage. So you do have to work on this before you do anything else investigating disease and particularly outbreaks. This is a really important one. Establishing disease causation is difficult, and this obviously doesn't just apply to wild animals, it applies to the whole spectrum, humans and also domestic animals. And also the fact that infection is very different from disease. So they're absolutely not the same thing. And this example is sort of comprises both of these challenges. This was one of the last koalas that I autopsied during my PhD. So that was around maybe 2016. And we have the stomach here open. This will be the pylorus, which is that junction between the stomach and the small intestine, and this will be the duodenum that is open now. And you see these little creatures here, they shouldn't be present. Those are tapeworms. So koalas very commonly are infected by a tapeworm that is called Bertilla obesa. I found them in about 30+% of the koalas in my PhD. And most of the time they were doing nothing to the animal. These animals were in perfect body condition, just happened to have been killed by a car because it was breeding season. They were moving around. So it doesn't really mean that, obviously, if I see this in my dog, I have a greyhound at home, I would definitely want to treat her. I'm not going to wait until see what happens. But in wild animals, they are an ecosystem by itself, and I'm sure you are very well aware of that. So we don't really have to go and try to treat them as if it was a domestic animal. What I did observe incidentally is that the koalas that had a lower body condition tended to have this higher number of the cestodes. So it became this sort of chicken or egg situation. Are these animals, because they were, because they were of a low body weight for whatever reason, did that maybe change slightly the microbiome? Many changes to dysbiosis. Did that potentially just lead to that disruption of the host-parasite relationship? And now these creatures are proliferating. Or are they actually acting like an actual parasite and taking nutrients from the host? It's not, I didn't answer that with my PhD anyway, but very common infection in koalas. So these are just some of the challenges of working with wildlife. But so some of you may have maybe thinking, is it really worth, despite all these things that you've mentioned this morning? And obviously the answer to that on my side is absolutely. I find it fascinating to overcome, all of these challenges, and there's many reasons for that. So an obvious one is we want to investigate cause of disease and death in not in koalas, in just general wildlife. But a really important one, especially now in the context of climate change, is the fact that we can actually detect early emergence of pathogens, and some of those pathogens may have a relevance for zoonotic, potential, or for wildlife conservation. So important example of this condition, of these challenges, of these issues, is American crows started dying in super high numbers in the United States in the 90s, and the relevant authorities collected these animals. They did a postmortem examination, and they detected the first arrival of West Nile virus in the country. So American crows became an unwilling sentinel for the infection, and so obviously every time that we see these massive mortalities in wildlife, we do want to do those exams and find out what's happening. Flying foxes in general, bats are a really diverse, taxa. They carry, that diversity in their species is also reflected in the amount of pathogens that they carry. And so we know that flying foxes are a host for henipaviruses, which are Hendra virus and Nipah virus. Hendra virus has been really relevant for our situation in Queensland. Hendra is not really far away from here, where it was first described, and we do autopsies in horses in the hospital. So we always only receive horses that have been either vaccinated or have been excluded by PCR from the infection. So every time, and also, the big one is Lissa virus. So every time that there's a mortality of flying foxes, obviously it makes a lot of sense to collect those animals and under a BSL-3 level lab to do, the right type of examination. But probably one of the highest impacts of veterinary pathology and doing autopsies is conservation. With those tools in veterinary pathology, you can monitor rescue, recovery, reintroduction and translocation efforts. You can also collect data, to see if any changes that you've done to mitigate a particular thing or changes in husbandry, if they had any impact or effect. And it can even give you indirectly some support for establishing protected areas. And just gonna talk a little bit more about that. I had the pleasure of working with Julia Creek Dunnards. I don't know how many of you know these little guys, but they're super cute, and they live in kind of like in the maybe middle to north Queensland in a very small area, sort of endangered marsupials. And there was an effort to create a captive breeding program some years ago in this in Gatton. And they did it, and some of them did reproduce, but unfortunately a lot of them did not. And they ended up reaching senescence, which in a Julia Creek Dunnard is quite fast, between 2 to 3 years. So they started developing these really amazing lesions that were all just sitting in individual reports in our database. So I decided to compile all those cases and be like, what happens? What should we expect in any type of, conservation breeding program attempt? Like, what happens if those animals reach geriatric age? What sort of lesions do they develop within that conservation context? And that's we found these really, lots of cystic lesions in the reproductive tract of females, and the males tended they had some lymphomas that were affecting the skin as well. So it was a really interesting thing. And that citation is, that paper was published this year. In regards to the establishment of protected areas, so all of you will have heard that koalas and chlamydia are unfortunately a thing. And the prevalence of chlamydia varies across mainland Australia, but we do have populations of koalas, at least until I checked recently, that are still free from that infection. So right here in this little area, that's Kangaroo Island in South Australia, that population is still free of chlamydia. So if you, in a way, can keep ensuring that any animal that died, if they get a postmortem exam and you can still verify that they are free from the disease, you can actually use this, information to influence legislation, protect their habitat further, impede habitat encroachment, and safeguard that species and that particular area. And maybe you can even use some of these individuals to repopulate areas that have undergone local extinctions, if that's the case. So we already went through what clinical pathology is. So that's the area of pathology that deals with bodily fluids. So that's also super useful. I don't work with that. It's a very different type of training, but it's also fascinating. And it really helps you to establish these baseline parameters. So I'm gonna go back to my time in Georgia to talk about this. I worked in a project with avian hemoparasites, and we were sampling over 50 bird species across all 4 seasons, and that included migrants and resident birds. And one particular bird, and it was this one. So this is indigo bunting. This is the male, that's the female. And you can't really see the size, but they are really tiny. Probably the heaviest is gonna be at 20 grams. So it's a very small bird, super pretty. But what was interesting is that we kept doing these, we were taking samples and also swabs. But the samples were to do these blood smears to have a look at the morphology of the cells in the microscope. But we were doing this microhematocrit too, which is this little tiny, very fine tube that as soon as you put it in contact with blood, it sort of like sucks it in. And you're able to spin it, and that's how you get how many red blood cells, white cells, and how much fluid is actually in that sample. So you can get a very quick estimation of how the bird is doing. And it's a very simple test. So we kept doing that for all of the birds, but consistently the indigo bunting had this really high number of red blood cells. And it was to the point where any other animal would be either dehydration, so it was severely hemoconcentrating, or maybe super stressed, it was contracting its spleen and was liberating all of these red blood cells into the peripheral circulation, or maybe could it have a really weird disease like polycythemia. Anyway, we didn't want to go there, but we were like, it has to be a very simple explanation to this because it was a statistically significant result. So once we had that result, we went into the life history of the bird. And this is a long distance migrant. They go from North America and they travel all the way, they cross all the Caribbean Sea, and they go into their wintering grounds in North and South America. So that's over maybe 1,500 kilometers that they have to travel. So they have this extremely high demand for oxygen carrying capacity. So that was influencing this change. And so if we didn't know that, and we didn't have the ability to put that in a paper and publish it, if an animal like that is taken to a veterinarian and then they do this test and they realize, oh, it's 55, 60%, red blood cells, this animal is dehydrated, and I'm going to try to correct that. It wasn't necessary. So that's when clinical pathology comes in. Obviously, quite an important aspect of veterinary pathology is forensic data. So we do work with principal authorities in the vet school, and we do, these forensic animal examinations. You can detect, potential cases where there's been welfare issues or neglect, and that mostly involves domestic animals, but it could also involve wildlife. And obviously, we're a teaching and training institution, so we work with undergrads, HDR students and specialty training students with autopsies about that. And once you, I work with both Bachelor of Veterinary Technology students and Bachelor of Veterinary Science students. And when they reach that point of doing dissections, a lot of them are really excited because this is one chance to actually see a body with lesions of these really classic veterinary diseases that is not the same as sitting in a textbook or a photo. So the students become really adept at case solving, and they keep working on their critical thinking and diagnostic skills by doing dissections. So because I just talked about training and being in a training institutions, I compiled this data that I'm going to show in the next slide because I wanted to know, well, I'm in the vet school, how much are actually our students getting exposed to wildlife subjects? Because in Colombia, in my university in particular, I just had 2 electives in 5 years. So that was, and everything else, if I wanted any training, it was up to me to basically volunteer at zoos or something like that. So UQ is nowhere close to that. We have a really incredible diversity of courses. So our 2 flagship programs are the Bachelor of Veterinary Science and Bachelor of Veterinary Technology. And for every single one of these 2 programs across all the 3 years for Vet Tech and 5 years for the Bachelor of Veterinary Science, everyone is getting exposed to both wildlife animals and exotics. So I was so happy to see this. So it goes from nutrition to pain management, surgery, anesthesia, welfare, and even legislation. So our students are absolutely getting a very good training, and I'm quite jealous that they have access to this, and we don't in such a diverse and mega diverse country like Colombia. So hopefully it's going to change. So this is the end of my talk. Sorry, I went a little bit over 30 minutes, but some of the references that I utilized, and this is the end of my presentation. Is there any chance that we can go into the Padlet? This is an actual link. If we click to, is it possible? Oh, really? Yeah. Otherwise, I can see it at home, but obviously I prefer to see it here. Check Chrome.
What's that?
You might be opening Chrome already.
Let's just, oh, there we go.
Oh, yay. Avian influenza, yeah, that's a good one. So small things on I like the simplicity of that, yeah. Scary bad things. That's true. Study diseases that can affect human and non-human ways of the ecological interactions between them. Fantastic, yeah, absolutely. Important to consider due to it representing animal well-being and welfare, enables identifying and treating disease. Yeah, that's very nice. And how disease spreads. We can absolutely use. So, thank you so much for participating. You know way more than hopefully, and what I was thinking that I might get. So, thank you for that.
Really? That's way more than you were.
That's what I mean. You have they summarize it all, right?
Thank you very much. Any questions?
I was talking to Alison Peel, you probably know Ali.
Yeah.
And she was talking about the lorikeet and the flying fox paralysis and something around botulism. Have you been doing tox studies on that?
Yeah, so unfortunately, yeah, so what we have access to, and we have spent like $5,000 in testing. We sent samples of those lorikeets to Western Australia because they're the lab that have the botulism PCR, and we ended up detecting 1 positive lorikeet, but it's to the bacterium, not to the toxin. So results are irrelevant. And so unfortunately there is and I can't say the name because it's this very long name that I've never seen in my life. But the University of California is developing with the toxicological team at the actual Davis branch. They're developing a test that doesn't require the bioassay, which is the mouse test. So what we do is that obviously mice get injected with the toxin, and then you just see the symptoms. That they have this flare chest, and they become paralyzed and semi-paralysis. and then you can give them an antidote and you stop the process. But that's a gold standard, which is obviously carries some welfare concerns. So we're trying to move away from that, and hopefully next year that test was going to be ready. That's the last time I heard about it. But botulism has been on the list for a very long time. It's just that it's very frustrating to actually find that toxin. And it's not easy to do that bioassay in Australia. So California does it, and it was done in the lab when I was a resident. And so that's why we've been thinking about potentially just saving some samples and sending them there. Not to the extent of what I know. I don't know enough about botulism to say yes or no, but we met with Dr. Francisco Sal, and he's the botulism expert, or actually general Clostridium expert, in veterinary diseases in the world. So he's the one that told us that it was either that new test that they were trying to develop or the bioassay. That's the gold standard. Yeah.
Another question. If nobody else does. Research students, are you getting many research students from the vet school? Do they actually go further than like in the wildlife space I'm talking about?
Not from the vet school. I've had now 2 master's students. One comes from the School of the Environment. She graduated last year, and she's the one, but she was a veterinarian that just happened to have an interest in wildlife. And we worked on that project about the role of koala retrovirus in bone neoplasms. And then this year I'm working with an AGAFES, which is the School of the School of Agriculture and Food Sustainability. Used to be called SAFS, now it's AGAFES, and she is looking into the spatial temporal distribution of those lowercase Prowse syndrome cases in the last hotspot. And the idea is that she's going to also do some surveys around the hotspots, building like a little buffer around them to see if we can start sampling vegetation and also entomological fauna. Yeah, but not the vet school is not common. I do have undergrad, a couple of them that are interested in wildlife, but not as HDRs or postgraduate coursework.
Is there still an honors program?
There is, I've never had a student from an honors program, but there is that possibility too.
There are other wildlife vets, so you potentially get honors students, or is this just?
Yeah, so we have in the small animal hospital, we have Professor Bob Donnelly, and he's the primary clinician that helps him, Mark Owens, and both of them do have those students sometimes, and most of the people sort of like think about them, not really think about pathology when they want to do wildlife, obviously, but I get a lot from them too. So I've worked with them in at least 2 projects thus far. Yeah.
Interesting.
From anybody from wildlife science, are those ones that you are getting from the School of the Environment? Are they in the wildlife science program?
I believe so, because it was a master in conservation biology, but then they did that research to become a master of conservation science, if I understand it correctly.
Yeah, that's a different program, but that's interesting.
Okay,
That's
yeah.
actually a lot of the students that come to CBCS and T's into like our conservation science or conservation biology.
Yeah, it just happens that the student that I got, she visited Hidden Vale Research Station, and then she saw the ad of my project because they funded that project, so that's how she got into contact with me. So, and she was a vet, like I've never had a non-vet contacted me to do research in wildlife pathology or wildlife diseases. That hasn't happened yet.
Questions online?
Not yet.
Just posting yes.
Hi, really interesting talk. I'm doing a similar kind of topic in my PhD at the moment, so I'm looking at expert perspectives on integrating welfare into conservation decisions. I was wondering if you could tell me a little bit about if there's any like particular challenges you found with kind of merging those 2 disciplines. Have you found like conservation was quite receptive to integrating or considering those like the studies you're doing?
I will say yes. I just can't think of a particular example right at the moment, but I at least, for myself and the people that I always work with, welfare is an issue. We want to obviously use the least number of samples, that we need to confirm something. So that's one thing. And obviously once you apply for an ethics permit, it kind of like puts you into that, mindset, and you start thinking like, how do I make this project the best way welfare possible? It helps in a way, the fact that when you work with dead animals, there's a very separate, it's not a full ethics application, it's this separate, like it's called ANRAFA, which is a ethics application not require a full ethics application sort of thing. It's a very long name, but it's become like ANRAFA. And so it tends to be easy, but obviously you need to know how the animal was either put down or was it found dead, where did you get it from, who was handling that animal, like all of those questions are also part of that process. So yeah, it's definitely a concern, but I can't commence my work in wildlife unless I have a, either an unwrap or athletics. Yeah.
Thank you.
Yeah.
In terms of like, if you find a whole bunch of dead animals, what is actually the best way to preserve them for pathology? Like, is it best to put them in the fridge, or is it a timeline kind of thing?
I guess the first thing is that you contact someone and ask them and explain the situation. If they're obviously very rotten, we can still do some things, but not as the whole spectrum of sampling. But if it's going to take, and this is not honestly like a hard rule, but it's like just to give you a general idea. If it's going to take over a week to get an animal to a particular place to do a proper autopsy, we say just freeze it at -20. If it's like a really a larger animal and you just happen not to have that freezer space, then at least if you put it on under refrigeration, and then just the animal can get to us as fast as possible. If it's something, I wouldn't encourage this in the field though, because we don't know what killed the animal, but obviously field autopsies are a thing. Obviously you need to talk to a veterinarian first before starting opening something that just died, because you don't want to do that, absolutely not. And I'm not going to encourage that whatsoever, but it is, those will be like the sort of the best type, and it is done all the time with livestock, for instance, that's a very common thing, but I will say over a week, freeze it, less than a week, fridge ideally, and then just get it as soon as possible to us or to Biosecurity Queensland, which is the other place that does autopsies in wildlife.
So I just want to ask, so I mean, like, if you come across like a dead animal, dead group of animals or something like that, who's their sort of like first point of contact? Like, do we like talk to maybe like local like vet clinic or something and bring it there, and then do they contact you, or do we, or like how do you know like when a big outbreak is happening?
I guess it depends on the situation, but I think Biosecurity Queensland will be a great place to start. They're very quick, responding to emails or phone calls. So I think that's probably just because you don't know what you're facing, and if you're facing something that can be transmitted to humans, you definitely don't want to deal at all with those animals. You just want to bag them up and send them somewhere. We have also received those animals, and we have the ability to do a little, let's say like with birds, because we're concerned about things like chlamydia psilocybe, which is a zoonotic pathogen. We can do those birds that we don't know what they died in a safety hood. So we have that ability, but obviously they have to be smaller animals that can be put in there. If it's something like a dingo, or like you saw like 100 dingoes died in front of you, I would definitely go to Biosecurity Queensland. So they definitely the first point of contact, and they have Anita Gordon is one of the pathologists there. She's been working with wildlife for a very long time. I go to her for questions. She's a very knowledgeable person. So yeah, that's what I would probably start. We just got an actual email, and I was, because I'm here, I can't do them, but 5 echidnas were found dead, and they were being submitted today for autopsy. So I asked somebody else to please do them, and then I'll take over the cases. So like in those cases, like we're not expecting something like anthrax, to kill 5 echidnas. It could still happen, but it's unlikely. So for those things, we can do those animals in a little safety hood, and then just obviously to be careful with ourselves. And then we take our samples. Everything gets washed properly in a different way than doing it outside on the floor. Yeah, so that's what probably start. They have a hotline, I believe, in Biosecurity Queensland for to direct you with questions.
I have a question regarding the roadkill animals. Or there is some study that reveals that already sick animals with some pathologies are killed more often on the roads than not sick animals. In a sense, like if animal is sick, is more probably they're gonna be numbed or something, then they can get struck by a car. Or if there is a relationship between roadkill animals and pathogens, by comparing the ones that die on the roads and outside the roads.
I am not aware of a particular study, and also I guess it just depends on the species, and you will have to do, I guess, a single study for every species. But I can talk about my experience. So when I did my PhD, I had two big projects. One was doing a prospective collection of data, and that was receiving koalas that were being euthanized since I started the PhD. And the other portion was an epidemiological analysis of data entry for over 20,000 animals over a span of 17 years. So in combining the results of those two data sets, like very roughly, it was very common for male koalas to be killed healthy during breeding season, which is really unfortunate. These animals were getting to me in perfect body conditions, really nice weight, like super nice looking fur. They just crossed my road at the wrong time, yeah. So that was a reality with koalas, and it was devastating, yeah. To see.
I'm asking because in São Paulo, Brazil, we had this yellow fever spread like some years ago. And the monkeys that were found that the road killed, most of them, I think 8% of them had yellow fever.
Oh.
So I don't know if they are already sick and trying to, I don't know, in bad conditions and get striked because they do not can climb or something like that.
Yeah.
But no one investigated if into the woods they are healthy or not. If it was like a general thing or just in the borders of the road or the ones that have been killed was mostly the ones that are sick.
Yeah,
I don't know, because
it speaks to the challenge that I mentioned, right? Like infection versus disease, because it could be absolutely fine that they are, they're just living their lives, they just happen to be infected, but they're subclinical and they remain subclinical forever. Or maybe they remain subclinical and then something else hits them and then now they have two conditions affecting them. So it is difficult to establish something is diseased, but that's why we need to do multiple samples. We examine all organs. If an animal has been killed by a car and it's very obvious, we still sample absolutely everything. And you need to do an autopsy of all, representative sections of all organs to be able to come up, hopefully, with an answer like that. Yes, this animal was diseased by yellow fever. So, ideally, you will want to see if it's a virus, you want to see inclusions, viral inclusions in cells, in the relevant cells that those viruses like to infect. And that's a very small window, unfortunately. So if you don't have the inclusions, it doesn't mean that the animal is free of disease. And that's when, doing PCR and collecting those extra samples helps. If you do serology, you could also have, responses in titers, and you can differentiate between an acute exposure versus a convalescent phase. So there's a few tools that you can use, to determine if they're disease or not. Or it becomes very complicated in a lab setting. You will probably want to fulfill what we call in pathology fulfill Koch postulates, which means that you have to get a pathogen or whatever is causing disease, put it in an animal, then the animal becomes sick in the way that you want it to become sick and show the right type of clinical signs. Then you isolate that pathogen from that animal and you put it somewhere in another animal and it replicates the same. So like that's how you theoretically prove that, yes, this is a disease causing agent. It has animal welfare, obviously implications and all that. But yeah, that's why it's tricky. It's tricky and you can't just do pathology and sit in the microscope and you have all the answers. You definitely need more than that.
Any other questions? Any online?
More quiet online.
Okay. Well, please join me in thanking Viviana one more time.